Hey friends, welcome to my channel. In this video, we will cover the topic assisted reproduction technologies. So, it is the reproductive technology that is used specially in the infertility treatment, and it is used to attain pregnancy through the uses of the procedures such as IVF, in vitro fertilization, artificial insemination, and the surrogacy. So, infertility can be defined as the inability to conceive after more than 1 year without use of contraceptions called as the infertility. So it can be seen both in the male and the female. Infertility can be seen in case of male due to several factors like sperm count disorder. Low sperm count includes oligospermia and the azoospermia. Some sperms can die, cells dies before reaching the age, and one of the major causes aged male have lesser sperm count. So the structural abnormalities such as the motility or the shape of the sperm, and in the case of the female it may be the inability of the ovaries to produce egg known as the ovulation disorder. We can say that the OHSS, ovarian hyperstimulation factor, quality of uterine lining such as the endometriosis damaged to the adjacent reproductive tissues, structure of the fallopian tube such as the blockage in the tube, and the maternal age also affects the infertility. So collection and the preservation of the gamete. So generally spermatozoa are preserved with the aim of maintaining their motility for the fertilization of the oocyte in vitro. And the standard method of the gamete preservation is the cryopreservation method. The cryopreservation required solution containing very specialized cryptocryptotech tanks that must store in liquid nitrogen at minus 196 degrees centigrade. The sperm fertility can be maintained even when the spermatozoa are stored without the specialized treatment for the preservation. And in one study, the fertilizing ability of spermatozoa was maintained in a solution adjusted to high osmolarity by adding the sodium chloride without adding cryoprotectants. So liquid nitrogen is normally used for the cryopreservation of spermatozoa and other cells, and although it is essential for the long-term preservation of the samples. So the preservation can also be done through the tris buffer and the EDTA, that is ethylene diamine tetraacetic acid. So cryopreservation of the human gamete and the embryo is an important and widely used method in the most embryology labs. So the types of the assisted reproductive technologies include the in vitro fertilization, as we have already seen in our previous videos. Next one is the gamete intrafallopian transfer, that is ZIFT. The zygote intrafallopian transfer, that is GIFT, and the intracytoplasmic sperm injection, that is ICSI. So ICSI has been applied as a powerful art technology in various animals including human. So the sperm cannot penetrate the outer layer of egg for a variety of reasons, and the egg's outer layer may be thick or hard to penetrate, or sperm may liable to swim, and in this cases the procedure of ICSI can be used along with the IVF to help fertilize the eggs. So in this, a single sperm is injected directly into the cytoplasm of the egg, and in this process a tiny needle called the micropipette is used to inject a single sperm into the center of the egg before it is transferred to the uterus. During the ICSI process, it may have some problem, like some of all the eggs may be damaged. The eggs might not grow into an embryo may stop growing. So certain conditions have been associated with the use of ICSI, such as the Beckwith-Wiedemann syndrome, Angelman syndrome, hypospadiasis, or the sex chromosome abnormalities. So next one is the GIFT, that is gamete intrafallopian transfer. So in this, it is another reproductive technologies of assisting pregnancy, which includes in removing the female gamete, that is eggs and sperm, and mixing it into the fallopian tube immediately, and it is a fallopian tube transfer rather the petri dish. However, a healthy fallopian tube is very important for the gamete implantation. Process of the gift. Firstly, the patient first needs an X-ray to make sure that at least one of the fallopian tube is healthy. A laparoscope is tubed to remove the eggs from the ovary. When the egg is taken out, the man provides a sperm sample. Next, the egg and sperm are mixed in catheter, and the mixed sperm and egg will put into the fallopian tube through the catheter. A medicine will be provided to women to help to build the endometrium and help in the plant the fertilized eggs. Next is the ZIFT, and it is a case of the IVF, that is in vitro fertilization, which is taken place outside the body, and the young embryo inserted into the uterus. This is a figure of the art process. Firstly, the eggs are aspirated from the ovary, then it is placed in the prepared semen sample, and the third step is the fertilized embryos or zygotes are transferred into the fallopian tube. So this is a figure of uterus. It is the ovary, this is a fallopian tube, and this is the cervix of the female. So it is all about the assisted reproductive technologies, and in next video we will discuss each of the part of the process of assisted reproductive technology. Thank you and keep watching.